Hi, my name is Roni Bar. I'm a PhD candidate at Professor Danny Michelson's lab. And today my talk will be about the effects of ApoE particle from various sources and lipidation state on the transport of cholesterol out of astrocytes and into neurons. I have no disclosures. Okay, so ApoE is a 299 amino acid long protein, and it is the main lipid carrier in the brain. There are three isoforms of ApoE, ApoE2, ApoE3, and ApoE4 that are differ by one amino acid in positions 112 and 158. Population studies revealed that among the afflicted Alzheimer's patients, more than 60% carry at least one copy of ApoE4. We can also see that ApoE4 lowers the age of onset of the disease in 7 to 9 years. The green line represents people with no ApoE4 allele, the yellow line represents people with one copy of ApoE4, and the pink line represents people with 2 copies of ApoE4. Okay, so when we examining the lipid content of ApoE molecule in human CSF, we can see that the lipid content of ApoE4 homozygote is significantly lower than the heterozygote and non-ApoE4 carriers. Another method to examine ApoE lipidation is through native gels. The gels contain no SDS and keep the native form of the molecule. In ApoE3 homozygote, we can see higher molecular species when compared to ApoE4 and heterozygote. So our conclusion was that ApoE4 is hypolipidated in humans. Following these results, we turn to examine ApoE lipidation on our lab model. And as we can see, ApoE4 is hypolipidated also in targeted replacement mice. Okay, so in the central nerve system, ApoE and astrocytes secrete ApoE and cholesterol through the ABCA1 transporter, and this process is referred to as cholesterol efflux. This cholesterol and ApoE complex is being absorbed through the LDL receptor and LRP1 receptor into neurons, and this process is referred to cholesterol absorption. So our hypothesis is that cholesterol efflux from astrocytes and absorption into neurons is affected by ApoE isoform and lipidation state. So considering the difference in the lipidation state between ApoE4 and ApoE3 and the role of ApoE in cholesterol delivery into neurons, we established an assay to investigate the effects of the ApoE isoforms on cholesterol release and absorption into neurons. We first seed immortalized human astrocytes in 96-well plate for 48 hours. Second phase is that we wash the cells and load body fluorescent cholesterol for additional 24 hours. We wash the cells again and add apolipoproteins as cholesterol acceptors. After 24 hours, we transfer a part of the efflux medium to a new 96-well plate and read the fluorescence intensity in the cell's medium. So up till now, we described the cholesterol efflux. The second phase of the assay is the cholesterol absorption into neurons. We incubate the same efflux medium on differentiated neuroblastoma cells. Cells are lysed after 24 hours and the fluorescence intensity is being measured, which represents the cell-bound cholesterol. Okay, so in order to investigate the effects of ApoE3 and ApoE4 on cholesterol release, we first used recombinant ApoE in rising dosages of 1 till 20 micrograms per ml. The control represents efflux medium with no acceptors, and the results are normalized to the control group. So in recombinant ApoE at dosages 1 till 20 micrograms per ml, we did not observe differences between ApoE isoforms in the control group, But past results at our lab determined cholesterol efflux with recombinant ApoE with much higher dosages of 40 till 160 micrograms per ml, suggesting that in recombinant ApoE dosages of 1 till 20 micrograms per ml are not sufficient to obtain cholesterol efflux. So since recombinant ApoE is lipid-free and ApoE secreted from astrocytes is present as an ApoE particle that have distinct properties from ApoE derived from other sources, we apply the assay utilizing astrocytes-derived ApoE. So here we use different dosages at the nanogram scale, 2.5 nanogram and 5 nanogram per ml, and we can see high levels of cholesterol released into the culture media in ApoE3 treated astrocytes. So the conclusion so far is that in cholesterol efflux and recombinant ApoE, we see little effect at dosages that are higher than 20 microgram per ml. And in astrocyte-derived ApoE, we see that ApoE3 is higher than ApoE4 at dosages of 2.5 and 5 nanogram per ml. So now we'll continue to the second stage of the assay, which is cholesterol absorption into neurons. In recombinant ApoE, we can see cholesterol absorption in both ApoE3 and ApoE4, with ApoE4 a bit higher and equal to ApoE3. And in astrocytes-derived ApoE, we observed a high absorption rate in ApoE3-treated neurons. And the conclusion here is that in recombinant ApoE, ApoE4 is higher equal to ApoE3, and astrocyte-derived ApoE, ApoE3 is higher than ApoE4, the same as the efflux. Okay, so the results so far has led us to further investigate and try to characterize how the lipidation state of ApoE3 and 4 affects cholesterol efflux and absorption. And we did it by separating ApoE particles that were secreted from astrocytes into different densities utilizing sucrose density gradient ultracentrifugation. And we did it as follows. 4 ml of astrocytic conditioned medium was loaded onto a sucrose gradient containing sucrose in different densities, stacked on top of each other, and together we have 12 mLs. We then subjected the gradient to ultracentrifugation for 48 hours. After 48 hours, 12 fractions were extracted. So this technique exploits the fact that in suspension, particles that are denser than the solvent will sediment, while those that are less dense will float. So as a result, higher density equals more protein and fewer lipids. Accordingly, fractions 1 till 6 represents the highly lipidated ApoE, and fractions 7 till 12 represents less lipidated ApoE. Best results of the 12 fractions detected ApoE mainly in fractions 4 till 9, so these are the fractions we examined. ApoE immunoblood of the examined fractions revealed that ApoE3 levels are higher than ApoE4, and most of the ApoE is present at the lipid-rich fractions, which are the low number fractions, 4 till 6. The different fractions were diluted to produce a constant concentration of 2.5 nanograms per ml. So in cholesterol efflux, we can see that in the low density fractions, ApoE3 promotes cholesterol efflux compared to control in ApoE4, while at the heavy density fractions, ApoE4 presents higher levels of cholesterol efflux compared to control in ApoE3. So these results suggest that the extent of lipidation is very important in cholesterol efflux. So the conclusion here is that in highly lipidated astrocytic ApoE, E3 is higher than ApoE4, and in less lipidated ApoE, ApoE4 is higher than ApoE3. So when we continue to cholesterol absorption, we can see that ApoE3 fractions absorbed well into neurons, and this ability diminishes in the heavier fractions, while ApoE4 levels remained unchanged. Okay, so the conclusions from the absorption is in both highly lipidated astrocytic ApoE and less lipidated ApoE, ApoE3 is higher than ApoE4. And so far, the overall conclusion is that in cholesterol efflux, the isoform specificity is determined by the extent of lipidation of ApoE. And I would like to take the opportunity to thank my amazing lab members, my PI, Professor Danny Michelson, and especially Dr. Ori Liraz, whose help was pivotal in our successful research. Thank you.